Hello again, everyone. Now we are in the topic of nutrition. This is a two-part session. The first part is going to be talking about some chemistry. Okay, very basic chemistry, but it's good to at least get this under our belt to understand the next section, which is going to be nutrition. Okay, and part of the digestive system and how it all works. Okay. So let's go ahead and start off with the chemistry of life. So here is, on this particular slide, we're just looking at the basic elements that are common to biology: carbon, hydrogen, nitrogen, and so on. And what these particular elements help create in terms of molecules will be the big four, your macromolecules, your carbohydrates, your nucleic acids, proteins, and lipids, okay? And this is basically where we need, these are the natural resources that our body needs, okay? And then here is a slide that has a list of the macromolecules, and then breaking each individual macromolecule down. The protein is created by amino acids. Then you have different structures and such. And then the carbohydrates, as we'll talk about, we have monosaccharide, disaccharides, oligosaccharides or polysaccharides. And then you got your different types of lipids, fatty acids, triglycerides, phosphoglycerides, so on and so forth. And then the nucleic acids, which are nucleotides. This is what makes your adenosine triphosphate, which we're going to talk about with cellular respiration. This is what's necessary. This is why we breathe, believe it or not. It's not just to get air into our lungs, but it's one big chemical reaction. And then, of course, RNA and DNA, which is the coding system of how our cells replicate and how we create proteins so our body can function and do what it needs to do to survive and be efficient. All right, so just a basic, very, very, very brief chemistry lesson is the building blocks of molecules. Excuse me. Matter is, of course, occupies space and mass. Matter is composed of elements. Those elements are made of atoms, and then, of course, we have. You don't have to know the periodic table, but I know you've seen this before. Is we have a periodic table. We have various acronyms or initials to reference, and they have different characteristics and so on. So, of course, you don't need to memorize that, nor do you need to know it in this particular class, which is nice. So let's just make sure we understand a little bit about atoms and what comprises an atom. Here, of course, is a water molecule, and then this is your oxygen. This is the nucleus. And of course, this is a hydrogen, and this is the nucleus of hydrogen, okay? And then it does look like Mickey Mouse here, so there is a reason why that example with water, why they're on polar, basically one is on one side, one is on the other, forming 180 degrees. But there's reasons behind that, which we really don't need to focus on, but it is based on electrons, positive and negative charges. Here's just a nice little illustration that just shows that atoms, you put a variety of atoms together, you get molecules. And the reason why you need to know that is because there is a hierarchy, just like we talked about in the last section. We start with molecules or atoms, then molecules. Molecules, when they start to interact with one another, they form cells. Cells form tissues, organs. We start going through that hierarchical, organization chart, tissues, organs, organ systems, and so on. Then we have the organism. Okay, make sure you are, for the next couple of slides, that you definitely want to make sure you know this. The anatomy of the atom is the proton, electron, and neutron. Okay, again, very simple stuff. Inside the nucleus, we have some protons and neutrons. Proton has a positive charge, P for positive, neutron is neutral, it has no charge, but it does have mass. And then we have an electron which dances outside the nucleus. It has a negative charge, electron, electricity. It's the flow of electrons that enables us to power stuff, okay? But it is so small it does not really have mass. And we don't really know, it doesn't really occupy one particular space. It's dancing so fast, around the nucleus. But as for illustration purposes, this would be like a hydrogen where you just have one electron and one proton, okay? So that's your basic anatomy. So we know that we got a positive and a negative because this is what has our mass, okay? Protons and neutrons, mass of 1, okay? Electron has no mass. So here's something that you want to make sure you understand. I don't think it'll be in any of the homework assignments, but this particular slide, in particular, you want to understand and appreciate, okay? This is part of the periodic table. So all that I want you to make sure you understand is what each particular box for an element means within the periodic table. Super simple stuff. Of course, the C for carbon is the symbol, okay? You've got your atomic number. What is an atomic number? Make sure you know this. It's equal to the number of protons in the nucleus. So carbon within the nucleus has 6 protons. And here's just a illustration for that. Of course, you have the name, okay, carbon. And then you have the atomic mass. Well, what is the atomic mass? Well, it's the average of the isotopes. We haven't talked about isotopes, but basically what it's saying is that there is a variety of different carbons. You've heard of carbon-14 or carbon dating. It just means that the number of neutrons can change depending on the age of that carbon, okay? So basically you got 6 protons, 6 neutrons on average, but when they add up, when you add up all the different isotopes, different variations of carbon, if you will, then the atomic mass is 12.011. Yes, it should be straight up 12, but because there are other variations, that number is going to fluctuate a little bit, okay? So that's all that you need to know about this particular diagram, which is a snippet from the periodic table, okay? So I don't think that's on the homework, but just do make sure you understand that. Of course, we know that all elements have different properties and different states of matter, whether it be liquid, solid, or gas, whether it be at room temperature. So here's an example. Copper is a solid, okay? And of course, copper is used because it conducts electricity and heat very nicely, okay? Now, if we wanted to melt it, I mean, you're looking at 1,000 degrees Celsius, pretty darn hot, okay? And these, these are just some of the physical properties. Stuff you don't know, but I just put in there for giggles and grins. All right, we can skip the chemical bonds. We don't need to worry about that. You can take, if you want to know more, take Intro to Chemistry. We don't need to worry about this one as well. All right, now let's look at water. Okay, water is the universal solvent. And what's important to understand is the characteristics of hydrophilic and hydrophobic, okay? So when a substance readily forms hydrogen bonds with water, it can dissolve in water and is referred as to hydrophilic, water-loving, okay? And then hydrogen bonds are not readily formed with nonpolar substances. Again, you don't need to appreciate those terms at this moment, but these are like oils and fats, and these are hydrophobic or water-faring. So what does all this mean? All right, something we're all familiar with, hydrophilic illustration here. Majority of us, if not all of us, have cars, okay? It's been a while, you haven't waxed your car in a while, all of a sudden you don't see the beads as you do here on the right, but on the left-hand side you just see blocks of water on it. What is happening is that the water is actually bonding with the paint. We do understand and realize what happens after a period of time if you don't protect your paint job, putting wax on it so the water does not bond with your paint, is you end up with rust. It starts eating away. There's a chemical reaction occurring. This is what's considered hydrophilic, water-loving. Okay, so we've seen that on the car. Now, if you do have like say you buy a new car or you just did a wax job, you see that the water beads. There is no reaction occurring with the paint. the wax or oil, okay, it's actually interfering with the reaction forming between the water and the paint, okay? And then that would be described as hydrophobic. So what's happening is that it's called the spheres of hydration, where basically the water molecules are having to bond within themselves together, and that's what forms that bead. It's a very weak bond. You know this because you can go in the pool, or we drink water and throw it up and you get droplets of water, so they break apart very, very easily. So hydrophobic, hydrophilic, okay? That's the big picture. That's what you want to take out of it. Don't worry about polar, non-polar. We're not interested in that. A couple of things. We do this actually in lab, is how water stabilizes temperature. So we're all familiar with boiling water. So you've got your water molecules, even without boiling water, just having a bowl, a pot of water, the water level will eventually drop. If you have aquariums, you know this. You always have to add water, whether it be fresh or salt water. And that means that the water molecules are escaping. There is energy within that water, whatever, even at room temperature. Okay? So that's called evaporation. You may not necessarily see it. You do see it as you add energy to a system. So boiling water, that means your water molecules are definitely moving around more so, right? And then you start seeing the escape through the surface tension, and water molecules are evaporating. They're escaping. And they're escaping in droves, if you will, because you see it, right? Because the air temperature is cooler, so the water molecules are coalescing a bit, and then you see it as vapor or evaporation, okay? And then on the right-hand side here, we have liquid water. We just have the regular molecules. Of course, the yellow is the oxygen, and the little green balls are going to be hydrogen. And then as we take energy away, so say this is at room temperature, and they're just bouncing around, being happy. And then as we take energy away, they're going to start coalescing, forming a lattice structure. And you know it's a lattice structure because those of you that live in northern climates, or even here, we don't see, snow as an example. You see it more so with a crystalline pattern than, say, in ice cubes. But this is what forms is your ice, okay? And there's no energy. The lesser the energy, the slower that the molecules are moving, and you form more of a solid, okay? Everything I just said. Now here's something that's kind of neat. This is more FYI. So we've got, again, I mentioned that water is an excellent solvent. It's a universal solvent. Solvent meaning that it dissolves stuff, okay? So we've put salt in water. You make pasta, you add salt as an example. Or you have salty water. You've tasted salty water like you gargle, I think if you have a sore throat or something. I don't know what the witchcraft is. Anyway, so here is a salt molecule, or a salt cube. It is cuboidal, by the way. You've done this if you've had salt water, lots of salt in the water, and you let the water evaporate, and you end up with these salt crystals or squares. It's super cool, if you're bored and just want something entertaining to do. But anyway, but here is the chemistry behind it. So sodium chlorine, that's an ion, okay, positive and negative charge. Sodium is positive, chlorine is negative. But check this out. So what happens is, as these dissociate, when you put the salt in the water, the chlorine and the sodium dissociate, okay? They separate, they break apart. And what's happening is your water molecules, look at this, you've got the negative charge of the chlorine right here in the bottom left, and then look at what's happening with the water molecule. You've got the positive charges of the hydrogen facing in. There's a reaction, there is opposites attract, so they're forming a sphere around the chlorine ion, okay? And now look at sodium. Likewise, you have a positive charge, but check it out. There's more of a negative charge on the oxygen, so the hydrogens are facing away because there's no attraction between the hydrogen and the sodium because they're both positives, right? So you have a sphere of molecules, of water molecules, surrounding the sodium ion. And of course, when you taste salt, you're tasting the sodium and the chlorine. It gives you that sensation of saltiness. Okay. Anyway, this is just illustrating how the concept is. Make sure you realize water is an excellent solvent. Okay. All right. Next thing is, again, we're talking properties of water, cohesiveness. So we talked about that hydrogen bonds. Okay, when hydrogens bond together, it's a very important bond in chemistry or in biology. It's like rubber bands. Okay, you put a rubber band on one finger and put a band on the other finger, and moving it back and forth. You pull it apart too much, and it will break if they're skinny rubber bands. If you put multiple rubber bands, it makes it harder. That means, as an illustration, that's a stronger bond, and there are stronger bonds. Hydrogen bonds are the weakest bonds, but they're strong enough to be cohesive where you can walk on water if you're careful, like this insect here. Okay, so it's surface tension is what that's called. Okay, now of course, you can put a paper clip. People do it as tricks and so on, but of course, if you break that bond, then you're going to sink. We cannot walk on water. Because we would break those bonds. Okay. Now, we see it in nature where if there is, if nothing, this is an illustration of water not being able to bond. That there are certain plants, as an example, that have a waxy film on it. Right? So there's no bonding effect between the water and the plant. So again, the water beads, kind of like, or it's exactly the same thing with your paint job and your wax. Okay, that's all this is illustrating here for you. Okay, so that's all of water. The next thing, the only thing that we need to understand here is when it comes to pH. All right, your pH scale is 0 to 14. Make sure you know that. 0 to 14. 0 is very acidic. 14 is very basic. Okay, 7 is neutral. That's pure water. So if someone says, well, it's high acidity, that means that it's 0 or 1 or 2. If it's highly basic, it's 14, like Drano or liquid drain cleaner or bleach. Okay, that's basic. Now, all this means is the higher that we go on the scale to 0, okay, so we've got, like here at water, 7, we got a bunch of H2Os, right? Water molecules. But dancing within that liquid water, you also have hydrogen ions and OH ions, right? So you're drinking water, but as you go higher in the pH scale or greater in acidity, all that it means, or what's happening, is that you're having more H's, more hydrogens than you are OHs. So as a ratio, so let's go to urine or saliva. You have water, you have some OHs, but you have, the ratio is greater in hydrogen. Okay, this is a log pH, so it's a considerable amount, but nonetheless, there are more hydrogens. Okay, so saliva, believe it or not, is acidic. Coffee, rainwater is acidic. So again, more hydrogen, even more hydrogen. So now where you're getting like battery acid, lemon juice, yes, you have water. Yes, there is a little bit of OH, but majority of it is in hydrogen in liquid form. Okay, that's what the pH and acidity is. Opposite end of the spectrum, we go from pure water to seawater. It's salty, but it has more OHs than H's. Baking soda, there's even more OHs than H's. It's just opposite. Milk of magnesia has more OHs. Ammonia, so on. Okay? So understand that, you win. You understand pH, the scale. That's all you need to know. Not the chemical reactions and all that. That's for chemistry if you wish to understand that. All right, now lastly to this is understanding the biological molecule. And we have our lipids and how they're important. We have amino acids, which are polypeptides, aka proteins. We have our carbohydrates, and we have our nucleic acids. They're DNA, RNA, and ATP. So let's take a brief look at each one of them, okay? You're going to have to know these big four. Now to begin, and all of them share these, is the carbon-carbon bonding, okay? So we are known as a carbonated species. Why is carbon so prevalent? Well, the reason being is that carbon has 4 electrons on its outer shell. Bottom line, it can bond, it wants to bond and can up to 4 different elements, okay, especially hydrogen. So this right here is an example of a stick model, just illustrating that this is carbon and it's bonding with hydrogen on all 4 spots. It is full. So as an example, CH4, that's methane. Okay. And then you can grow chains from this. So you can take this hydrogen off and add another another carbon, and have some H's, then add another C and then some H's, and on and on and on and on and on, as you'll see in just a second. Okay. All right, the first macromolecule, carbohydrates. Okay, carbohydrates. So we've got carbon. So this, when it comes to carbohydrates, notice that this is an illustration of a ring. Okay, each angle is a carbon. Carbon here, carbon here, carbon here, carbon here, carbon here. Except for this little guy, we've got an oxygen. You don't need to memorize that. I'm just pointing this out for those of you that aren't familiar with this kind of stuff yet. What this is called, the first type of carbohydrate is a monosaccharide. Mono meaning one. We've got your glucose, we've got our fructose, and we've got our galactose. Okay? Our body, when we eat food, of any type of food, we are trying to break it down into its simplest components and ultimately get to what's called glucose. This is what our body needs to make ATP, that makes us do what we do, okay? We want to break things down into its main components. So the food, all the macromolecules are packaged into that McDonald's cheeseburger, into fruits as fructose or sucrose with sugar, all sorts of, whatever. But ultimately, our digestive system's goal is to break it down into glucose. And that's what our body can use, is glucose. Okay? This is just illustrations of, understanding. This is a chemical formula or a molecular formula. Make sure you know what this is. It's a 1-to-1 ratio, 6 carbons, 12 hydrogens, and 6 oxygens. Okay? So this is glucose, C6H12O6. Make sure you know that. We don't need to worry about this. This is just showing different illustrations of glucose and so on, but don't worry about that. All right, so that was a monosaccharide of carbohydrate. Then the next one is going to be called the disaccharide, di meaning 2. Okay, so as an example, if I add 1 monosaccharide of glucose and we're able to bond it with another monosaccharide called fructose, we end up with the form of sucrose. And you know what sucrose is? That's table sugar. Okay? And so what happens is that when we take in sugar, then our body wants to break this molecule down into glucose. Okay? It'll break it down to glucose and fructose, but what our body uses is glucose to get into our cells so that there can be a chemical reaction to make ATP so we can yawn, run, roll our eyes, talk, whatever, repair ourselves, so on and so forth. So these are disaccharides. And disaccharides, this is just an illustration. So we've got maltose, which is two glucose molecules. We have sucrose, which is going to be glucose and fructose. And then lactose, which is found in milk. Some people are lactose intolerant, is glucose and galactose. Do not need to memorize these, but just you'll see them, especially if you're in lab, you'll see these a few times. We'll go over those. You'll get used to them. All right, then the third type of carbohydrate is the more complex one. The most complex is going to be polysaccharide, okay? And that means many. So all it is that you got a bunch of monosaccharides that are linked together, right? So we can take, here is, a glucose molecule with another one, another one, another one, another one. You get the idea. It's a chain. And that would form what's called starch. Okay? We have another one, some different pairings, which forms glycogen. And then we also have cellulose. Okay? Now, just a couple of things regarding starch and glycogen. So starch, those are forms of sugar in plants. We find that in pastas, but you also find it in roots and seeds. Glycogen, we find that within our own bodies. Okay. And then we have cellulose, which you find in cell walls of plants and so on. Wood stuff is filled with cellulose. Okay, it's what gives it that rigidity, that strength. Like when we buy lumber, it's filled with cellulose, as an example. Okay. So this is where, when it comes to starch, you know what's starchy. Potatoes, macaroni or pasta. Corn is very starchy. So we have a problem with, sometimes breaking down some of these foodstuffs. Okay. Okay. I had to pause there for a second. I had to answer a question. Okay, here is a nice illustration breaking down your carbohydrates. Monosaccharide, disaccharide, polysaccharide. Okay, so these are the names of your monosaccharides: glucose, fructose, and galactose. And then, again, don't spend so much time you don't need to memorize this necessarily. Just have an appreciation of what mono, di, and polysaccharide are. Just know, if anything, know what your monosaccharides are. Realize that sugar, sucrose, is a disaccharide, meaning it's comprised of two monomers. And then polysaccharides are many monomers attached together, like starch. Okay, what's important to understand is that what and how they can be broken apart or how they can be bonding together to create such molecules. Okay, and we'll be getting into that very shortly. Okay, the next macromolecule is a lipid, or fats. Okay, these are hydrophobic, water-fearing. Okay, and your various lipids are triglycerides, phospholipids, glycolipids, and steroids. Okay. And so we'll be talking about a few of these. Again, we're not going to spend too much time on it. What I do want you to appreciate is the anatomy of a fat molecule. Okay. So here is, we have, as an example, here is a glycerol. It is a molecule right here. And then we've got these fatty acid tails. Okay. And then we go ahead and have a reaction. Okay. And then we end up forming a triglyceride. Now, triglycerides, for those of us that go to the doctor or have more doctor specialists than friends, like me apparently, is that if you have too many triglycerides, that's not good. It's part of your diet and all that. But what happens is that if the body can't process or have areas where these fatty acids can go, then what happens? They get stored into adipose tissue and adds to your fat. Okay, they have calories. They're not going to be used, so it gets stored in fat. Or adipose tissue, that's what fat is, which we learn about that in lab as well. Okay? And when it comes to different fats, I think there might be one question on exam that has this, just realizing there's saturated fats and unsaturated fats. The difference is that saturated means it's saturated with hydrogens and they're single bonds. Typically, these are going to be solid at room temperature, like your animal fats. You know, if you cook a nice steak like a ribeye and, you know, you're full, and then after, 30 minutes, you'll notice that fat starts forming because it's cooled off and it becomes solid. That's why you can't just eat that type of fat continuously on a daily basis, because yes, your body will thin it out and get rid of a lot of it, but there's a point where you get super saturated with it and it does start clogging your arteries, and it's just exactly as you see on your plate. Okay, it's solid fat in comparison to unsaturated fatty acids, which these you have double bonds. Okay. And these have a characteristic where they are liquid, like olive oil, as an example. They're liquid at room temperature, so the body can process it a lot easier, and it doesn't necessarily get attached to your linings of your veins or arteries. Okay. There's a lot more to it, but we don't need to get into detail. I don't want to get into detail of it. It's not necessary. There's too many other things we need to talk about. But anyway, those are your characteristics of saturated and unsaturated fats in a very nutshell. Okay, phospholipids. When it comes to fats, this is what our biggest player is that we want to focus on. Again, there's many others, but the phospholipid. Okay, why phospholipid? Because that's your main component for your cell membrane. Okay, so here, this is that terminology, polar and nonpolar. So the polar head, this is considered the head. All right, this is the glycerol, this head part. This is hydrophilic. I get confused from time to time. Hydrophilic. It's water loving, which is good because this is on the outside of the cell, right? So our bodies are made of water, so our cells are floating in water. Okay, then the tails, okay, you got two of them here. These are nonpolar. These are the fatty acids. Okay, just see the chains? Carbon, 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 and there's two hydrogens, one on top, one on bottom. These are hydrophobic, and these are on the inside of the cell. Now, what these phospholipids do, they create a phospholipid bilayer. Okay. So you got one on the outside, one on the inside. This is the one on the left here. This is facing the inside of the cell. Hydrophilic, water loving. It's this in-between area that is no man's land. There's a larger scale of it. Okay. Hydrophilic head, hydrophobic tail. Here's your cell membrane. Okay, you got your heads on outside and facing the inside, and the tails are on the inside. Okay. Now, there are things that can penetrate the cell membrane. There are quite a bit that cannot. Okay. So let's take a look at some of the players that we have. Very simple. We have channels. These are proteins, molecules that have formed this type of conduit that allows various molecules into the cell that are too large to fit in between these phospholipids. Okay, we have carbohydrates that are like receptors. This is how cells communicate with one another, because cells can be right on top of one another. So if there's a problem with one cell, the rest of the body will know about it. Okay, if you're ever wondering, how does the body know if I've got a scratch? Let's see, I have to pause very quick. Okay, that was a nice 35-minute delay there. Uh, okay, phospholipid membrane, protein channels, hydrophobic tail, hydrophilic head. I think that should be good. We'll be going across the. Okay, and then lastly, or almost lastly, are the proteins. Okay, and this is something that we're really going to be getting into in terms of protein synthesis, how they're created and such. Okay, proteins are very, very important in the body. This is why we have DNA. Okay, DNA is the code book, if you will, that makes proteins. Okay, proteins take on a variety of different forms. They're structural, regulatory, protective. They can help in reactions. They can be toxins, enzymes. Okay, a variety of things. Okay, so proteins are amino acids in a chain forming a polypeptide. Okay, so this right here, this top illustration, this is a polypeptide chain of amino acids. Okay, and then the protein can take on various shapes, and the shape dictates the function. Okay, so depending on what it's shaped as is what it does. Okay, it's quite simple. And actually, that's what the Golgi body does. It helps package that particular protein. And that's, again, the Golgi body is organelle within our cell. So we'll be talking about proteins in depth a little bit later on in the course. There's just another redundancy that I do in lecture class. Here's another chain of amino acids. Anyway. And then the last is going to be nucleic acids, deoxyribonucleic acid. Of course, make sure you understand what DNA is. And then we also have RNA, ribonucleic acid. RNA is the molecule that is created that transcribes the code from DNA for a particular protein and then gets translated outside of the nucleus by ribosomes that actually makes the protein. And we'll talk about that process in several weeks. And to me, it's absolutely fascinating, fun stuff. But we'll talk about that, genetics and so on. I really enjoy that topic. And then here is just giving you the anatomy of DNA. We have a nitrogenous base. We have a sugar and a phosphate group. So here we have, this is your nucleotide right here. Not necessarily something we need to know today, but we will have to know it eventually, so you can't avoid it. So you might as well at least take a peek at it now to understand it. So when it comes to RNA, this is a backbone. RNA is a single-stranded chain. Again, we'll talk about this later, but this is RNA. It's a single channel or a single-sided molecule, okay? And then DNA, as we're seeing, it's called the double helix. And so this gray portion is the backbone of, or the ribbon is the backbone of DNA molecule as well as the RNA. Okay, and this one piece of backbone plus its accompanying amino acid, or as well as your phosphate. This, what's within the square, is called a nucleotide unit. Okay, your nucleotide unit, and that exists in both DNA and RNA. Okay, and again, we will talk about that more in detail a little bit later on. Just other illustrations for you to peruse through. These H's, by the way, again, I said hydrogen bonds are very prevalent in biology. This is what holds this DNA molecule together, okay? Consequently, this is also what gives it that bending effect, so it has that double helix. These are also very easy to break in reactions, to break them apart or to unzip them. And we'll find out how that works as well when it comes to DNA replication. Here's another example. It's like a ladder. So here is a backbone of DNA. Of one side, here's the other side, okay? These within the rungs of the ladder, these are your base pairs, adenine, thymine, guanine, and cytosine. They only pair with one another, and we'll talk more about that in detail later. And of course, then these are connected via hydrogen bonds, which that tension gives it, then what's here on the right, that helical shape, okay?